very much, Dave. Good morning, everybody. Yes, I'm going to be the first one in a set of 3 presentations on eDNA, and we're hoping to highlight that if we talk about eDNA monitoring, there's quite a variety of different options, and it might not always be monitoring, might also be just an option to learn more about the ecology of the species. So the first talk, I'm going to basically give you an overall overview of eDNA, what eDNA is, and how it works in crown of thorns sea star monitoring. It's a joint effort between me, Kale, Maria Gomez, who's here, Jason Doyle, and Francis Patel, but a lot of other people are involved too. And so in the first section, I'm going to give you an overview of eDNA in general and some application, how we use it to quantify larvae and do larval monitoring for crown of thorns in the plankton. I'm going to briefly diverge from monitoring and explain why I think it's also an important tool to look back into the nutrient hypotheses and what other aspects on biology we can glean from monitoring larvae. And I'm going to give you the conclusions on an outlook at the very end. So this slide is completely redundant here. I suppose I don't have to say too much, of course, about crown of thorns because you're all experts in this field. As you know, it is one of the 4 species in the complex, and on the GBR we only have 1 species, which is probably Solaris. It's a native species here. It's not an introduced species, as some people assume. It's very fast maturing, as we've seen from Simon's data, probably spawning after 2 years. It's highly fecund and a broadcast spawner. So the 2 main hypotheses for outbreaks, as we talked about yesterday, are the predator release hypothesis, which is a top-down mechanism, or nutrient increase fertilizing larvae on the reef, which is a bottom-up. There's no reason why these can't work together in some fashion or work with other hypotheses to explain the outbreaks. Simon has shown this already, just because I'm in this talk going to talk about the larvae part. This is the typical larvae cycle. I think Maria has highlighted it too. We start from a standard very large egg cell, which is 180 microns big, which is quite big for a feeding larvae, and reach after several different larvae stages, a brachylaria stage at the bottom there, which is then settling on the reef, forming the juvenile. eDNA is just a very generic name for free DNA, which can be in the water column, it can be in sediment samples, in terrestrial samples. So the DNA can be free in the water column, it could be bound to sediment surfaces or absorbed to sediment surfaces, or also still be on cell fragments. In terrestrial and freshwater environments, it has been picked up for monitoring and diversity discovery over the last 10 years a lot. Not many people have used it in the marine world until about 5 years ago, because it was generally considered, the detection limits were considered an issue in the marine world. However, with increasing and much better technology these days, even those detections, as we will show you in the next 3 talks, are not really an issue. So it's been used a lot for detection of introduced species in freshwater environments, but also in sediment cores, identification of predators, of prey, in guts and feces of species is also a very common usage, and detection of extinct animals in sediment samples, for example, like from New Zealand, there's sediment cores documenting the death or the extinction of the moa, and similar cores exist from North America. I'm looking at mammoth. So you can do ancient DNA, and Kale has worked on that on the GBR in John Pandolfi's group before she came up to us. The genetic method is not prescribed. It's not one specific method, so most people might be aware of next generation sequencing approaches, which give you results on a lot of different species. It gives you hundreds of thousands of sequences, so they're very good for biodiversity descriptions. We work on single species detection with digital PCR. We initially used quantitative PCR, but basically we have a very specific primer. Hang on, just skipping a bit. We have a specific primer, as I'll show in a minute, just for Cots, and we can quantify the copy numbers of that primer, and we can work out what the number of Cots or the number of larvae is from that. So briefly, again, these are the three different topics we're going to talk about. I'm going to briefly mention the larvae. So all of these are obtained by actual plankton samples. We have one trip per year on an Ames research vessel where we have about 10 days collecting larvae in association with water quality parameters, which is again quite important for interpretation of the nutrients. But we also get more than 1,000 samples these days from the Cots control boat. So big shout out to you guys. We get an amazing support. We probably got the biggest plankton sample collection on the GBR by now. So we get a lot of support by stakeholders and by the control boats helping us to collect these water samples. Then Jason in the next talk is going to talk about eDNA to detect post-settlement Cots, and Peter Doll is going to give a presentation on settlement collectors. Not just using eDNA, but using eDNA as an additional method. Just briefly, as I mentioned before, the basis of all of this work for Cots was Jason and me developing about 6 years ago a couple of specific primers which just amplify crown of thorn sea stars. So we tested those against a variety of different echinoderms and other invertebrates and could show that only where crown of thorns is present we get positive detections. So in a very initial paper, we only, so this is the first trip we did in 2014 on the RV Cape Ferguson. We only did presence or absence because that was the first application. And we showed that in an amazing amount of samples in that year, we had positive samples. A couple of years later, we worked up the method so that we can actually quantify the larvae within a reasonable range, and we showed we had larvae up to 38 larvae per sample. That's the highest numbers we see here, which is still not a lot because a sample is, per cubic meter, because that's only 38 larvae in a cubic meter. What's important about that to me is that we use, if we're running experiments on larvae ecology, we're using much, much higher densities. We're using 1 to 2 per milliliter. So that has an implication for the interpretation of data we produce in the lab, but just only a sideline here. This is a presentation. Every individual dot here is one sample occasion. So as I said, most of the dots are samples we got from the Cots culling teams. All 5 boats are currently collecting and helping us a lot with this. So every blue dot is actually a negative, so there's no Cot, but every green dot is a positive detection. On the x-axis here, we just have time, and we have latitude over here. So this is the same data we looked again before. In 2014, we had nearly every sample positive, with big numbers of larvae in there. And coincidentally, I just talked to Simon. That is the year where Simon had the big spawning. So maybe there is a correlation where they all spawned in one go and hit. It might also be the end of the really huge outbreak numbers. We don't know that. You see, we get more and more samples also from the south over the time. We still get a good number of positive hits. It's generally getting a little bit lower this last year, so this is 2020. We did not analyze this year's data yet. We also finally got samples from down south, which we're really happy about. We find a quite surprisingly high number of positive samples in the Capricorn bunker group. You see, it's not many dots, but more than half of them are green, so it's actually quite a high number of positive samples down there. We also see a lot of samples coming up here. So basically, we get all those samples from John Brewer Reef and Keeper Reef now, from Pacific Marine, which is a fantastic data set which forms a new time series. You also see this long data set here at Moore Reef, which come from Eric Fisher, who supplied all this data as a continuous monitoring, basically, on the Moore Reef pontoon, which is already happening. That's just to illustrate how many data we got. One thing we have done and published with this data is back to the Moore Reef data. We actually looked at the seasonality of spawning, so no big surprise, there's only larvae in the plankton in summer, so you never find any larvae outside the main spawning season. What we could do with this data set is confirm that the spawning really starts pretty much exactly when the water temperature goes above 28 degrees, which is good. That's usually just before Christmas. So the best guess, I think the nearest full moon to Christmas is my best guess for the main spawning time of Cots. But as we have discussed before, that can happen to a few different occasions. So this is just an example of how the data can be used. And just at the end, a very quick teaser on what we can do with the nutrient data. So during all of the field trips, as I said, we collect 3 different types of nutrient data. These are chlorophyll data from the IMOS underway system we got on board. So basically every dot is just a station showing our growth tracks, and the size of the dot represents the chlorophyll density. So we've got the underway system data. We have a CTD station at every plankton station, and we take actual chlorophyll samples and other parameters. So in the long term, we're hoping to relate those samples to the quality and quantity of the larvae in the plankton. We haven't got those data analyzed. Just a quick teaser. Looking at the chlorophyll data from the field, you see the chlorophyll guideline values are depicted here, and this is the area, but there's no data yet, wait. This is the area which was considered of chlorophyll values too low for growth of crown of thorns larvae. However, if you're actually looking at field samples, most of these are just around that range of 0.2 to 0.3, which is considered quite low. So we, as Maria pointed out, there's probably a lot of secrets in the larvae we still have to resolve. However, one I'm interested in, these are the chlorophyll data of deeper samples. So I'm really interested, so this is about 20 to 30 meter deep. There's much more food down there. We just have no idea whether the larvae go down there. So one of my main aims with the CSIP is to, for example, work out whether larvae can go down and exploit those depths, which would have enormous implications for interpreting the nutrient hypothesis, because we don't even know what drives the chlorophyll out there. That's possibly not even land runoff. That might be just upwelling on the shelf or anything. So that's just a little teaser on what we can do. That's a summary of the CTD casts we're doing, basically confirming the same trend. You quite often in summer get much higher chlorophyll values below 20 meters. We have started to work on this. We have bought a very expensive plankton pump, which Jason is setting up here, so we can now pump plankton at certain depth. It just takes an enormous amount of time because you need to pump for at least 30 minutes. You've got to lower it down, so we can only do like 5 stations a day, and a huge research vessel has to dangle around the reef with us doing that. So it's not a cheap exercise. Anyhow, I think it's worth doing it. Coming to the summary of my part of the presentation, yes, I think we're getting a lot out of these larvae samples. Again, a huge thank you to everybody helping to collect the samples. I think it's a very powerful tool to elucidate lots of factors unknown in the larvae ecology, especially large and fine scale temporal resolution, spatial resolution, and the temporal resolution. Also important, I think, the data on on larvae can later on be used to verify models for connectivity. It's a very direct way to log a connectivity between reefs to find the larvae. So mentioned understanding outbreak of understanding the causes of outbreaks, but basically it's, well, also I think an important monitoring team. I know David doesn't agree on that, but I still think it's currently one of the earliest early warning signals we can get, because basically I'm expecting outside the outbreaks we will actually find 0 larvae. So as soon as we find any larvae, at least we get an indication that there's something happening in the region and we can have a better look at the benthos. And that's me. Well, all the collaborators, thanks again. Fantastic to get your help. Thanks.